Thank you. Good morning. Before I dive in, I'd like to introduce my research team, myself, my research partner Kyle Fulgham, Audra Stevenson, and Chelsea Brown. They've been really integral in helping us with our research. Also, I'd like to extend a special thank you to Sigma Zeta. You guys are supporting the pursuit of knowledge, and we're very grateful for all the work you do, and a special thanks for the research grant that has contributed to our research this year. So thank you. Let's get a little review of organic chemistry. It's my favorite subject. It was my favorite class, and a lot of people dread it. But chapter 5 of my textbook, anyways, talked about stereochemistry. So what we're looking at here is a compound that the carbon here is called an asymmetric carbon, which is defined as a carbon that has 4 different substituent groups bonded to it. So you can see here that there's 1, 2, 3, 4. Now the mirror image of this compound here has a different configuration of these of these constituent groups here. So what we see is that this mirror image is non-superimposable upon itself. So using the Kohn-Ingold-Prelog prioritization rules, we can designate this left compound here as the S enantiomer, and this one here on the right as the R enantiomer. Now, one thing about these asymmetric carbons is that if there's one asymmetric carbon in a compound, it's considered a chiral compound. Chiral compounds express optical activity when you pass a plane of polarized light through this sample of this compound. This rotates it either to the left, levorotatory, or to the right, dextrorotatory, and we're able to measure the angle of that rotation, and this is called the specific rotation. So we found that people have been trying to estimate, considering what aspects of the compound are present, to estimate the angle of specific rotation that a compound might have. So individual contributions of different functional groups, where they are. Attempts have been made at this. This Condrew and his researchers weren't able to make a conclusive conclusion based on their research. But so far, the specific rotation angle is independent of the structure as far as we know, and we're unable to predict whether it is levo or dextrorotatory based on the structure. We also are able to assume that a branching of a compound, especially the ones that we're looking at, which I'll show you later today, a branching of the compound will decrease kind of the surface area of this compound. And based on that, we're going to assume that, the literature data that we have found demonstrates that a decreased surface area is correlated with a decreased magnitude of the specific rotation. So if this is true, here's the literature data. We have pentanetol and 3-methyl-butanetol. So let's just focus on these two here. You can see that the pentanetol has a 7.65 angle of rotation, and the 3-methyl-butanetol has a 5.34 angle of specific rotation. So let's look at these compounds here. The one on the left is the pentanetol. You can see it's more of a linear compound with a single branch of the hydroxy on the second carbon, whereas the other compound here has a branch, so it's more of a globular compound and a decreased surface area. So since the magnitude of the rotation here is lower, we can draw that conclusion that the surface area is associated with the specific rotation. So our hypothesis states that if we can assume that, if that correlation that we can draw from that one piece of data is true, that if we continue to synthesize these 5 carbon compounds, the 2-ols, and we synthesize the different branches of these compounds, and are able to isolate the enantiomer and measure its specific rotation, that we would find that the more branched the isomer, that the lower specific rotation angle we will find. Here's butanetol. This is the compound that we worked with primarily for our research. It's a 4-carbon alcohol. The stereochemical center here is on the second carbon. You can see on the left we have the S enantiomer, and on the right we have the R enantiomer. These compounds have a specific rotation of 13 degrees. The S enantiomer is dextrorotatory, and the R enantiomer is levorotatory. Now, one thing we know is that enantiomers we're not able to separate them by physical properties because the enantiomers themselves have identical physical properties. So in order to separate them, we can use a diastereomer. Diastereomers have these different physical properties, including aroma, dipole moment, most importantly, boiling point, solubility, and sedimentation rates, because we can utilize these properties, or the differences in these properties, to isolate one of the enantiomers. Once we've isolated the enantiomer, we can then measure its specific rotation. So in order to do that, our theory takes us to the idea of using Fischer esterification. So we're going to use a racemic mixture, which is an equal solution of both R and S enantiomers. The racemic mixture of the butanol and react it with lactic acid in an equilibrium reaction in the presence of an acid catalyst, and we will get the sec-butylactate ester and water. Once we have this ester, this is a diastereomer. It's actually a mixture of the R-butanol and the 2-butanol with the enantiomerically pure lactic acid. Since we know that we have an enantiomerically pure lactic acid, these two we know that this reaction will only yield 2 different diastereomers from which we can separate. So we prepared these diastereomers by simply adding the butanol to a mixture of the lactic acid in the presence of an additional acid catalyst. Then we distilled them. Now, if you remember that Fischer esterification process is an equilibrium reaction, so in order to not play with this equilibrium very much, we decided that it would be best to pump as little heat into our distillation as possible. So we conducted it under vacuum. We were able to use this vacuum manometer to measure our pressure inside the apparatus, and we got down to as low as 5 kilopascals. Atmospheric is 760 kilopascals. So using a simple water aspirator, we were able to pull a significant vacuum on our distillation apparatus that would lower the boiling points of these compounds and allow us to separate them more easily. So, assuming that we were able to drive the equilibrium as far as possible, in our distillation flask, we're able to have a mixture of the R and S diastereomers and water. As we heat these slowly under vacuum, we're able to pull off distillate number 1. Distillate number 1 is a mixture of the butanol, unreacted butanol, and water. This will drive the equilibrium to the right, and we know that there's an azeotrope here, so we're able to watch them come over together. And since butanol has a finite solubility in water, we can see layers in this distillate. So when these come over, we can see very distinct layers that help us know that we have this. This is the compound we pull off. The second distillate is one of the diastereomers. Now, I must say that this slide is not exactly accurate because we're unable to know exactly which diastereomer we pull off at the next temperature. So for the sake of this talk, we'll go ahead and assume that we're working with the R. This is the R-sec-butylactate here. So for the sake of the talk, we'll assume that we have the R diastereomer. As the distillate 2 comes off, we will be left in the original distillation flask with the S enantiomer. Once we've separated these 2 diastereomers, in order to measure this rotation of the butanol, we need to hydrolyze the ester bond to get back to just the pure alcohol. So we use a sodium hydroxide solution, and we're able to, through saponification, hydrolyze that ester group and end up with the lactate salt and the pure butanol, which has isolated our enantiomerically pure butanol. Once we've done that, we're able to measure the specific rotation in a polarimeter. This takes a sodium D light and passes it through a plane polarizer, so we're able to watch the angle of rotation and measure that with our polarimeter. We measure this rotation and then we divide it by the length of our cell, which is 1 decimeter, and the concentration of our solution, which since we're using a pure solution of enantiomerically pure butanol, we can use the density in that case. Once we measure the specific rotation, we can then calculate the enantiomeric excess. Now you'll notice that this enantiomeric excess equation uses alpha max, which is the alpha of the pure enantiomer. So we're able to find the butanol in the literature as well as the pentanol, and I should say that we have repeated this entire experiment with pentanol because it has a decreased solubility in water, so we get greater purity and greater yield. But since we know the literature value for that specific rotation, we're able to measure our enantiomeric excess, or how pure is our 1 enantiomer solution. Now, I've talked a lot about this separation, and I mentioned to you that we want to draw a correlation between the branching of the compound of the 2-ol, the 6-carbon 2-ol, and its specific rotation. So the scope of our research this year and last year has been just to simply establish this separation process. We've currently gotten to a 36% enantiomeric excess going through this whole process. So we know that we don't have a pure enantiomer. We're not measuring the exact pure optical rotation angle. So we're not able to synthesize our compounds and then go on and draw our conclusions based on that information. So moving forward, the first thing we're going to need to do is improve that separation process so we can obtain an enantiomeric excess of 100% and see that, okay, we know that our separation process works and we can isolate one of the isomers, or I'm sorry, one of the enantiomers of the 6 carbon tools that we plan to synthesize. Actually, I'm sorry, we purchased them with our grant money. Thank you again. So this is inaccurate, sorry. Synthesized isomers, purchase the isomers of, and isolate the 2-hydroxy enantiomers of the 6 carbon compound, which we'll see here. And you'll notice that we're keeping the first 2 carbons the same, the 1 carbon, 2 carbon with a hydroxy group, and just the side chains here that are changing. This is so that we can observe the effect of this branch and what effect it has on a specific rotation, leaving the independence of the hydroxy group. Once we've been able to isolate these enantiomers, we want to collect the specific rotation angle of that pure enantiomer, and then compare the numbers using our hypothesis and trying to decide whether or not the one compound with greater branching has a lower angle of specific rotation than that with less branching. Our whole goal is that we can essentially draw a correlation between the amount of branching in a compound and its angle of specific rotation. Now somebody might be asking, why the heck do you care about what this compound rotates like? I understand it's a confusing concept and why it's important, but let me tell you, we all know that enzymes are very specific, and sometimes an enzyme can only react with one enantiomer of a compound. Perfect example, ibuprofen. So unless you are taking an enantiomerically pure ibuprofen, only half of the ibuprofen in your body can actually be utilized by the enzyme. So that's just one of the examples. Once we're able to predict this angle of specific rotation, it can help people understand whether or not they're able to separate the enantiomers for medical compound use. So I'm going to pull this slide up again because this is where we're at mainly in our research, is the distillation. But I'm going to stop here and ask if there are any questions. Yeah.
You said you were able to compare to the absolute value found in literature for the compounds that you're trying to like determine the purity. Is there, for all of these different branching ones that you're looking at, is there literature that's already been shown like what their specific rotation is, or?
We really hoped that answer would be yes, and our project would be done if that were the case, because we could just look up the numbers and draw the conclusions ourselves, but we haven't been able to find literature data for all of those isomers, just the 4 and 5 carbon alcohols, which we were actually able to get from the vendor. So we were presented with that problem when we first wanted to do this as a literature research. We found out that that data has not been collected, which is why we've proceeded to actually collect the data ourselves.
Okay, so then how do you determine like the excess for those other ones?
We're using the 4 and 5 carbon alcohols, the pentanetol and the butanetol, which we have the literature data for, in order to make sure that our process of separation is right. And then we're going to repeat that process with the other alcohols in order to separate them and hope that we can draw conclusions off proper data that way. So, good question. Thank you. Anybody else? Yes?
D and L-glucose, are they fit into something similar to this?
Yes, they do. The D and L nomenclature is a biochemical nomenclature. We're using organic chemistry, which I know they're very close, but the Kohn-Ingold-Prelog rules have to do with the prioritization of the groups, and that's how the naming is derived. The D and L compound that you mentioned is based on the way the structure is drawn out. I can't remember. The Fisher projection, I think, is what that's called. The way that the Fisher projection is drawn out, from that they derive the nomenclature to decide which.
It's how it relates to glyceraldehyde.
There you go. So I know that the D and L letters, which are not dextro and levorotatory, which is confusing, that I was talking about earlier, they're actually different, though they're biochemical nomenclature. Yes?
Could you remind someone who hasn't taken organic chemistry in a few years what a diastereomer enantiomer is versus an enantiomer?
Yes, I will. I'm actually going to back up here and try as fast as I can to get to a slide that helps illustrate that pretty well. What we're looking at here is the enantiomerically pure butanol. And for those of you who don't remember from organic chemistry, the second vertice here on this compound is a carbon, and it has 4 substituent groups bonded to it. On the left is a methyl, on the right is an ethyl, a hydroxy on the top, and then what's not drawn is an assumed hydrogen. So there are 4 different substituent groups on that particular carbon. This is known as an enantiomer, considering it has the opposite stereochemical arrangement. So you can see that these are mirror images, and they're non-superimposable upon each other. Now from here, we can then look at these are the 2 diastereomers. You'll see that one of the carbocarbons, which is the one on the lactic acid on the left here, it has the the methyl, the other big group on the right, hydroxy, and then the hydrogen. And then the other stereocenter, or the other asymmetric carbon on the right here that is contributed from the butanol, is not necessarily pure. We have a mixture of the 2 of them. So I have a diastereomer here defined as stereoisomers that are not mirror images. So these 2 compounds aren't mirror images of each other, but you notice that the butanol in the previous slides are their mirror images, but you cannot superimpose them on each other. It's like using your hand. They're mirror images of each other, but you can't wear the right glove on the left hand. So that's a good question. It's really hard for me to explain, and it was hard for me to understand the concept too. But the diastereomer, they're non-mirror images of compounds. Thank you.
You're missing a second OH on the bottom.
Yeah.
Correct?
I think so. On the bottom.
Your hydroxy is missing on your bottom.
Right?
No. The one on the top shouldn't be there. My bad. Thank you. Yeah, this is the one on the top here should be a butanol, but the O coming off on the top left here is what has been esterified with the lactic acid. So the one on the bottom should actually be a CH3, or this group right here should actually be a CH3.
Okay.
My software screwed up when I was drawing these out, and I had to do it 6 times, and it was a mess. And so, sorry about that. Thank you for pointing that out. Any other questions? Awesome. Thank you so much for your time, and again, thank you so much.